Good afternoon. My name is Ty Martin. Viruses such as HIV and H1N1 affect millions of people worldwide, and it is of critical importance that we find new biomedical technology to fight antibiotic-resistant bacteria and viral mutation. And today I'll be talking about a computational study of viruses and their interactions with a unique class of compounds studied by the Witten group here at UNM. So first I'll give some brief background of virus structure and the structure of the compounds in this study. And then I will also discuss docking simulations involving the binding of these compounds to virus surfaces. And from these results, I will then discuss molecular dynamics interactions between these two. So briefly, I will talk about the structure of both the virus capsid and also our molecules in this study, and followed by the antimicrobial and antibacterial properties of these compounds. So this is a structure of one of the compounds used in our simulations, which we call OPEs. And you can see that there are positively charged side groups here and here. And also on these molecules, N represents the number of repeat units. So you can have polymers of these molecules. Next is a picture of the structure of the virus used in our simulations, which is called MS2. This virus is 27 nanometers in diameter and has 32 pores around the outer capsid or protein coat. And this protein coat is made of 60 biological units surrounding genetic material, which is shown here in red. And it has been shown that the OPEs have antiviral and antibacterial properties. In this slide, we see some results taken from the Witten group that show, on the left, living bacteria in red, which have been incubated with OPEs for 10 minutes. And after an hour of incubation with OPEs in the presence of UV light, you can see that all of the bacteria is dead, which is indicated by the green coloring. These molecules also have been shown to have antiviral properties. In these images taken by Ying Wang of the Witten group, you can see on the left are several MS2 capsids that are fully intact, and you can see a clearly defined shape and boundary. And on the right is after an hour of incubation with OPEs, and you can see that the boundaries are clearly deformed. And the proposed mechanism for why this occurs is that the OPEs coat or cover the outer capsid surface, and in the presence of UV light, you can see that the capsid is clearly damaged. And this leads us into my current research objectives, which is to find the most probable binding sites of OPEs to the capsid surface, and to explore the interactions of OPEs and viruses using molecular dynamic simulations. For our docking simulations, we use a software package called AutoDock, and that allowed us to see the probable binding sites of the OPEs to the virus surface. And from those results, we also wanted to see what the charge distribution was at those binding sites. So if we focus on this region indicated by the red boundary around this pore, we can see from the docking results that there are OPEs binding to multiple sites around the pore. And in particular areas, there are higher concentrations of OPEs bound to the surface. And this is a more probable or likely binding site. So from this, we wanted to see what the electrostatics or the charge distribution on the surface would be where these higher concentrations are found. So in this image, we see an electrostatic surface with red indicating negative charge and blue indicating positive. And the OPEs that are currently binding in that location are indicated in green. And what we actually noticed is that the positively charged side groups on these molecules orient and are attracted to the negative regions of the capsid surface. And from these docking results, our next step was to use molecular dynamics, which will allow us to see or determine the interactions between OPEs and the capsid surface. And this is a still image of our simulation system. And you can see the OPEs beginning to bind to the surface of the capsid, which is indicated in tan. And if we take a look at an animation or video of this simulation, can see the OPEs beginning to move and interact with the capsid surface. And there are also ions present in purple, and the smaller dots are water molecules which have been reduced for clarity of the video. And from further analysis of this, we found that negatively charged amino acids seem to interact with the positively charged groups on the OPE. So amino acid residues such as aspartic acid. And for both of these studies, we found that OPEs do in fact bind to more negatively charged regions around the pore on the capsid surface. And we also determined that the interactions begin to occur at 1 nanosecond. The only problem is this is not a very long time scale for us to see what the interactions actually are. So we're looking at two different methods to decrease the computational time it takes to obtain these simulations. And the first method is called coarse graining, which allows us to reduce the overall number of particles in our system. So on the left you can see what's called an all-atom representation, and on the right is the coarse grain model that we were able to generate. And you can see a clear decrease in the overall number of particles. And the other alternative that we're looking at is called Brownian dynamics. And this allows us to remove explicit water molecules from the system. This method is also superior to implicit solvent models because it does account for the viscosity of water. And with that, I would like to thank my mentors, Dr. Deborah Evans and Eric Hill, and also Dr. David Witten, and the other groups listed here. And here are my references used in the study, and I would be happy to take any questions at this time.
So Ty, when you look at the coarse grain model, what kind of molecules or atoms are you moving? How do you create a coarse grain model that still retains sufficient of the properties of the whole?
Well, there's actually a couple different types of coarse graining. In our case, we use something called shape-based coarse graining, and it basically reduces the number of atoms by 200 times. And each particle that you see in the coarse grain model is sort of an average of the properties of 200 atoms. So that's how it works in our case.
So you mentioned about the Brownian dynamic,
Mm-hmm.
superior to the implicit solvent. What exactly is it about how it models the viscosity of water that sets it apart from just having a dielectric implicit solvent?
Let's see. So that allows it to have an implicit solvent.
So what is different between just an implicit solvent model and the Brownian dynamics is there in simulation?
Well, the water or the viscosity would affect how fast the interactions occur, or how the molecules move through the system. Mm-hmm.
Do you have any data on the response of mammals to OPE?
As far as simulation data, or
Actual experimental data?
from some of the simulations or some of the data that I've learned about, that there have been some done on epithelial cells, as far as I know. But other than that, none that I know of at this time.